Hello everyone, welcome to my channel Ardent Flower. Today let us discuss about photosynthesis in higher plants. This is part 1. So before we dive into the topic of photosynthesis, we all know about autotrophs and heterotrophs. What are autotrophs? Auto means self. These are basically organisms that can synthesize their own food. They can synthesize their own food. What are heterotrophs? These are the group of organisms which depend on other organisms for food. So here we are studying about plants. So basically we know that plants are autotrophs. Not only plants, algae, they are autotrophs. Heterotrophs examples include humans, we are heterotrophs. Then we have animals and fungi. They are basically heterotrophs. Bacteria, if you consider, there are certain bacteria which are autotrophic, which can synthesize their own food, while few are heterotrophic. So, if you remember, we have already studied in grade 7 about photosynthesis, where we know that it is a process where light energy is getting converted into a chemical form, chemical energy. So, the raw materials required for photosynthesis are your carbon dioxide and water, where in the presence of sunlight and chlorophyll, the pigment, green color pigment in plants, giving rise to glucose with release of oxygen and production of energy. So the raw materials for photosynthesis are your carbon dioxide, water with sunlight and chlorophyll, and glucose is your end product, oxygen is your byproduct. Okay. Now there were few experiments which were conducted to see for the presence of starch. Now, I told you that plants are basically autotrophs. We have discussed it just in the previous slide, right? So, we know that they perform the process of photosynthesis, as a result of which they can cook their own food. And during this process of photosynthesis, we know that the end product which is formed is your glucose. The excess glucose, once the plant utilizes its need, the excess amount of glucose which is formed gets converted into starch. So to test for the presence of starch, there are two experiments which were conducted. First one was done with a variegated leaf. What is meant by a variegated leaf? When I say a variegated leaf, it means which is partially green and it is partially white. As you can see through this picture, it's green here, and so basically there are two regions, one here and two. Basically this region is green and this is white in color. So to test whether starch formation is seen in both these regions, what has been done? A drop of iodine is added in both these regions. What was found is this region showed blue-black coloration. Why is this blue-black coloration observed? This is because when light falls in this region, we know that the green color is because of which pigment? Chlorophyll. This chlorophyll pigment absorbs the light which has fallen on the surface of the leaf and performs the process of photosynthesis, where the carbon dioxide gets converted to glucose and excess glucose gets converted into a starch. Now the starch which is present It reacts with our chemical iodine to give blue-black coloration. But when we add a drop of iodine in this white region, what was observed is there is no coloration seen. Why is it so? This is because this region is devoid of chlorophyll, no chlorophyll. So even when light falls, it cannot be absorbed, as a result of which no conversion of your carbon dioxide into glucose and thus no starch formation. Since there is no starch, there won't be any reaction occurring and hence there is no coloration observed. So this experiment basically proves that chlorophyll is essential for the process of photosynthesis, right? Now let's see another experiment which was conducted. Here we have taken a leaf and covered only some part of the leaf with a black paper strip. Okay. Now let us consider this is region 1, region 2 and region 3. Okay. So when it is exposed to light, we know that light falls on the surface of the leaf. The chlorophyll which is present absorbs, as a result of which photosynthesis occurs. Right? When photosynthesis occurs, what happens? Glucose is synthesized and this glucose gets converted into starch. Excess glucose gets converted into starch. So upon addition of iodine in this region, what happens? Starch which is present, which is formed due to the excess glucose formed during photosynthesis, reacts with the iodine, resulting in blue-black color. Right? But when I remove this black paper strip and add a drop of iodine, there will be no coloration seen. Why is it so? This is because the black paper did not allow the light to be absorbed to reach the pigments present inside the chloroplast, as a result of which no photosynthesis takes place. So there will be no glucose formation and hence no starch formed in this region. Therefore, there is no coloration. Basically, chloroplasts are your cell organelles which contain your pigment chlorophyll. It is the site basically where your process of photosynthesis takes place. So now this experiment again indeed shows that sunlight is essential for the process of photosynthesis, and this experiment proves that chlorophyll is required for photosynthesis to take place. Now let us see an experiment to show that carbon dioxide is essential for photosynthesis. Now if you remember the equation of photosynthesis, we studied it as carbon dioxide, which is your raw material, along with water in the presence of sunlight, and chlorophyll gives you glucose, which is nothing but C6H12O6, along with oxygen and energy which is released. So basically this carbon dioxide and water are your raw materials. So we have seen an experiment to show that sunlight is necessary for photosynthesis. We have seen an experiment even for chlorophyll, which is this, without chlorophyll there will be no photosynthesis taking place. Now let's see an experiment demonstrating, showing that how carbon dioxide is essential for photosynthesis to take place. Now let's see this plant where the leaves are intact. With the plant we have not plucked the leaf. Now one side here, if you observe this leaf, it is placed in a conical flask. Basically the conical flask is closed with a cork, and the cork has some pores here allowing the air to go inside. So here the leaf is placed in a conical flask containing KOH. KOH is nothing but your potassium hydroxide. This potassium hydroxide actually absorbs your carbon dioxide. So what happens is, we know that this plant is kept in the atmosphere. So when air enters through the pores in the cork, carbon dioxide which enters is absorbed by KOH. So no carbon dioxide is available for photosynthesis. So there will be no photosynthesis taking place. But if we consider this leaf here, it is placed in a conical flask containing water. Okay, so carbon dioxide is entering, and this carbon dioxide, along with the light and water, the usual process of photosynthesis takes place. Now, what we do is take this leaf, both these leaves, leaf A and leaf B. Now, when leaf to the leaf A, when you add iodine, there will be no coloration seen. But to leaf B, when you add iodine, there is blue-black coloration. Color observed. Why is it? This is because in leaf A, though light is present, the air which is entering containing the carbon dioxide, it is absorbed completely by KOH. So there is no carbon dioxide left for the plant to take in for process of photosynthesis. As a result, there is no glucose formation, no starch, and hence no coloration seen in the case of leaf A. Whereas in case of leaf B, what actually is happening is light. There is presence of light, water is present, then you have the carbon dioxide. So the usual process of photosynthesis takes place, as a result of which glucose is formed and excess glucose gets converted into starch, and this starch reacts with iodine that we have added, resulting in blue-black coloration. So this experiment again proves that carbon dioxide is essential for the process of photosynthesis. So these are all the experiments that you have studied in your grade 7. It's just we have recapitulated uh, to show that how carbon dioxide is essential for photosynthesis. Chlorophyll, the pigment present in your cell organelles, chloroplast, is essential, and sunlight is. So these are few experiments to show that these raw materials play a very key important role in the formation of glucose, where excess glucose gets converted into starch, and this presence of starch is seen by addition of your chemical iodine. Now let's see. A famous scientist by the name Priestley has conducted an experiment where he has taken a bell jar, has taken a candle which is burning, and closed the candle along with the mice with a bell jar. So you can see here, basically, after some time, the candle stopped burning. Here it was burning and the candle stopped burning, and even the rat died. So in the first observation, this is his first case, and this is the second case. In the first case, you can see that the candle along with the mice were placed in a bell jar, and after some time, the rat died first thing. The second thing is candle burning was stopped, right? Just a minute. Candle stopped burning. More precise. Burning. Now he has taken another case where he has kept candle, mice, and to it, along with it, he kept a potted plant in the bell jar. To his surprise, what he has observed is the candle continued burning, and the mice survived. So, Priestley, after seeing his observation, he hypothesized that there is something in the plant which is there's some gas which is being released by the plant which is purifying the air in the bell jar, causing the mice to survive. But later, now we know that this plant is undergoing the process of photosynthesis, as a result of which the plant releases oxygen. This oxygen is utilized by the mice for its breathing, as a result of which it can survive. And even this oxygen helps the candle to burn, and therefore it continues to burn. So these were the two observations he has seen, and he hypothesized that there is something which is being produced by the plant, and this gas is nothing but O2 that we know now. And Priestley later named this gas as oxygen. So this was the experiment which was done by Priestley, where he has taken a bell jar with, in the first case, he has taken a burning candle along with the mice in a bell jar, and he found out that the candle stopped burning and even the mice died. This is because the candle, after it stops burning, it releases carbon dioxide. There is more accumulation of carbon dioxide, no oxygen here. So the mice died. No survival of the mice, and the candle stopped burning. In the next case, has kept a potted plant in the bell jar. The potted plant released oxygen, and this oxygen helps burning of the candle, and it also allows the mice to survive. It helps in the process of respiration, and hence you can see the candle being burnt and the mice survived. There is another experiment which was conducted by Ingenhouse, another famous scientist. Basically, Ingenhouse just modified the experiment of Priestley. Modified experiment of Priestley he has done. What he has done is he took a plant, an aquatic plant, hydrilla. He has taken two conditions where he placed the plant in the presence of sunlight. One case, that's the first case, and in the second case, he placed it in dark. No sunlight was given. Now, in the presence of sunlight, when he has placed this plant in water, we know that in sunlight, along with carbon dioxide and water, process of photosynthesis takes place, during which glucose is produced along with oxygen being released as end product. This oxygen could be seen as gas bubbles in the water. So these gas bubbles suggests the release of oxygen by hydrilla due to the process of photosynthesis. But when this plant was placed in dark, there were no gas bubbles seen. Why is it so? Because since there is no light energy given, as a result of which what happens, there is no photosynthesis taking place in the dark, or very little photosynthesis actually takes place in the dark. As a result, no air bubbles seen because no oxygen is being released. So Ingenhouse experiment again suggests that sunlight is essential for the process of photosynthesis. It re-emphasizes that sunlight is a raw material. It is also required for process of photosynthesis. So basically, we know the basic equation of photosynthesis, where carbon dioxide reacts with water in the presence of chlorophyll and sunlight to give glucose and oxygen with the release of energy. We have studied few experiments to show that carbon dioxide is essential for photosynthesis. Chlorophyll, sunlight are also very essential for this process to occur. Here we need to understand one thing, that C6H12O6, that is your glucose, is your end product. Means that is the product which is formed due to this reaction, whereas oxygen is called as your byproduct. What do I mean by saying byproduct? It means this product which is produced, oxygen, is not utilized by your plant. In turn, it is released by the plant as a waste product. So it's being utilized by humans and animals, where we use it for respiration. And excess glucose. See, this process of photosynthesis takes place until you give sunlight and there is amount of water and carbon dioxide present in the atmosphere. So always the process of photosynthesis continues to take place. So there will be glucose accumulation. So this excess glucose gets converted into your starch. So what is the storage form of glucose in plants? Storage form of glucose in plants is your starch, right? So in this part 1, we have just studied few basic experiments to show the importance of the raw materials for photosynthesis to take place. Let us meet in another video with few more experiments on raw materials of and their importance in the process of photosynthesis, and we also study about not only about chlorophyll but also the other accessory pigments required for this process to take place more efficiently. I hope you like this video, and if you have any doubts, please comment. Feel free to comment, and please like, share, and subscribe. There will be many more videos, even related to chemistry, that will be put up in this channel. Please subscribe. Thank you.